Good morning. Now, in continuation with the previous presentation, when we were comparing the structure of purines and pyrimidines, we find that the distance between the two polynucleotide chains always remains constant. And the reason behind that is the hydrogen bonding between the thymine and adenine and thymine, cytosine and guanine. So because of these bonds, the distance between the two polynucleotide chains of the DNA remains constant. Then we discussed about the central dogma of molecular biology, which explains that the information of the DNA is transferred through RNA and then it is converted into proteins. The detailed process of all this we'll be studying in the coming presentations. This proposition of double helix structure of DNA and its simplicity in explaining the genetic implications became revolutionary, and Francis Crick proposed the central dogma of molecular biology, which states that then genetic information flows from DNA to RNA and then to the proteins. In some viruses, the flow of information is in the reverse direction also, and that is from RNA to DNA. The flow of information also is reverses from RNA to DNA. And can you suggest a name of this process? This process is called as reverse transcription. We also call it as reverse transcription. Now, in the packaging of DNA double helix, when we take the distance between the two consecutive base pairs, we know that it is 0.34 nanometer. And if the length of the DNA double helix of a typical mammalian cell को अगर हम consider करते हैं and उसको simply calculate करते हैं, how it is done? It is done by multiplying the total number of base pairs with the distance between the two consecutive base pairs. So this is how we can calculate the length of the DNA double helix. So here we can multiply by simply multiplying the total number of base pairs with the distance between the two consecutive base pairs. We can carry out this process. Now here the distance between the two consecutive base pairs is 6.6 into 10 to the power 9 base pairs, and when it is multiplied by 0.34, that is the distance between the two consecutive base pairs, and then when we multiply it, it comes out to be nearly 2.2 meters, a length that is very far greater than the dimension of a typical nucleus. So that is why approximately 10 to the power 6 meters. So how is such a long polymer is packaged in a cell? So that needs to be looked into detail. So if the length of the E. coli, is 1.36 nanometers, so can you calculate the number of base pairs in the E. coli? So the distance between the two consecutive base pairs in a double helix DNA, if it is 0.34 into 10 to the power 9 meters, and the total length of the double helix in the DNA, if we know that it is calculated by multiplying the total number of base pairs with the distance between the two consecutive base pairs. So total length of the double helix DNA is equal to total number of base pairs, which is multiplied by the distance between the two consecutive base pairs. So this comes out to be, by multiplying, we'll get this value. So the number of base pairs in the DNA of the E. coli comes out to be 1.36 nanometer into 4 is, that is 4 into 10 to the power 6 base pairs. Now here we can see in prokaryotes such as E. coli, though they do not have a well-defined nucleus, when we consider the prokaryotes, we find that they do not have well-defined nucleus, and DNA is not scattered throughout the cell. So DNA, it is not present throughout the cell or in the nucleus. It is not scattered like that, but the DNA is negatively charged. Okay, DNA is negatively charged, and it is held with some proteins. Okay, it is held with some proteins. You can see here, these are the proteins, globular proteins which are present. So these proteins that have positive charge in a region which is termed as nucleoid, and the DNA nucleoid is organized in the small large loops which are held by proteins. So which are held by proteins, you can see it here. So in eukaryotes, the organization is much more complex. There is a set of positively charged basic proteins which are called as histones. These are the histones, and a protein acquires charge depending upon the abundance of amino acid residues with charged side chains, and histones are rich in basic amino acids residues which carry the positive charge in their side chain. So histones, they are actually the unit of 8 molecules which are called as histone octamers, and negatively charged DNA is wrapped around the positively charged this histone octamer to form a structure that is called as nucleosome. So you can see here, these are the nucleosomes, and a typical nucleosome contains 200 base pairs of DNA helix, and nucleosomes constitutes the repeating unit of a structure in the nucleus which is called as chromatin. This is packed in the form of chromatin, and these are the thread-like structures which are colored bodies which are seen in the nucleus. These are thread-like structures with colored bodies which are seen inside the nucleus, and the nucleosomes in chromatin is seen as beads on strings. So you can see here they appear as beads on string structure when viewed under an electron microscope. So these nucleosomes, so many they are present inside a mammalian cell, and the beads on the string structure in the chromatin is packaged to form chromatin fibers. These are the chromatin fibers, and here you can see the difference in the prokaryotes and eukaryotes as explained just now. In prokaryotes, we know that DNA being negatively charged, it is held with the proteins which have the positively charged, and they form a structure that is called as the nucleosome, and the structure is called as nucleoid. And the DNA in the nucleoid is organized in large loops which are held by proteins. Eukaryotes की अगर हम बात करें तो इसमें भी positively चार्ज्ड बेसिक प्रोटीन्स हैं जो हिस्टोन्स हैं. हिस्टोन्स ऑक्टामर की फॉर्म में प्रेजेंट होते हैं. नेगेटिवली चार्ज डीएनए उनके आसपास रैप अराउंड करता है, एंड दैट फॉर्म्स अ न्यूक्लियोसोम, एंड दिस फॉर्म्स द क्रोमेटिन. And theoretically, how many such beads, nucleosomes, do you imagine are present in a mammalian cell? Approximately 30 million nucleosomes may be present in the mammalian cells, and a diploid cell contains this much number of nucleosomes with the number of cells which are present. So the packaging of chromatin at higher level requires additional set of proteins, and that collectively referred to as non-histones proteins. So here we can see that the non-histone proteins in a typical nucleus, some region of the chromatin are loosely packed, stains light, and they are referred to as euchromatin, and the chromatin that is more densely packed, that is again genetically inactive, we call it as euchromatin, which is uncoiled or loosely packed, and that form a chromatin, and they are genetically active. So heterochromatin and euchromatin, they both appear like this. You can see here, this is the euchromatin. It is an uncoiled, loosely packed form of chromatin, and it is genetically active. And here you can find this is the heterochromatin. It is such part of the chromosome which is firmly packed, and it is genetically inactive. So when they are stained, they appear differently. They are darkly stained, they are lightly stained.